Hello everyone, welcome to today's session. Today we are going to see class 12 biology chapter 5 molecular basis of inheritance. In that we are going to see Human Genome Project. In the previous sections we learned that DNA is the genetic material and that the genetic information of an organism is determined by the sequence of bases present in its DNA. So in simple words, if we want to say the genetic makeup of an individual lies in the DNA sequence. So scientists thought if two individuals are different from each other, that DNA sequence should also be different at least at some locations. So this idea led to a very ambitious question. Can we determine the complete DNA sequence of the human genome? So in order to answer this question, the Human Genome Project, it was launched in the year 1990 and it was a 13-year international mega project and was completed in the year 2003. And what is the main purpose of this project? See, the main purpose was to determine the complete sequence of the human genome and also to understand the genetic information contained within it. So the project was coordinated uh, mainly by the US Department of Energy and also National Institutes of Health. The Wellcome Trust of the UK was also an important partner. So along with the contributions from countries such as Japan, France, Germany and China. And why was it called as a mega project? So the human genome contains approximately 3 into 10 power 9 base pairs. So that means that about 3 billion base pairs, right? So at the beginning of the project, the estimated cost of sequencing, it was about 3 US dollars per base pair. So how much it will be? Just calculate. So 3 billion, right? So 3 billion multiplied by $3. So approximately it will be, yes, US dollars, it is $9 billion. So the amount of information was also enormous. And if the DNA sequence from a single human cell was stored in a book, so approximately it could have been 3,300 books. Yes, and scientists they needed some powerful computers in order to store, retrieve, and analyze these huge amount of information. It says you can able to see all the important details here. Yes, so go through all these things. These are very important ones. So next we are going to see the goals and also who ran it, or we can say as like coordination of this Human Genome Project. So what will be the major goals of this project? First, identify the human gene and to determine the sequence of the DNA. Next is to store the information, then improve the data analysis, and next is to transfer the technologies and at last address the ELSI. So I'll explain everything now in detail. So the first one, identify all human genes. So it was one of the important goal, yes, to identify approximately 20,000 to 25,000 genes present in human DNA. So the actual estimate obtained later was it was around 30,000, but which was much lower than the earlier estimate. How much was the earlier estimate? It was 80,000 to 1,40,000 genes. Next is to determine the DNA sequence. Determine the DNA sequence. So the project it aimed to determine the sequence of approximately 3 billion base pairs. How much? 3 billion base pairs. So that make up the human DNA. We already know the nitrogenous bases present in DNA, ATGC, right? Adenine, thymine, guanine, cytosine. So the order of these bases, it carries the genetic information. Next, after finding it, we have to store the information. We have to store the information. So this information, the project, it was generated an enormous quantity of DNA sequence data. So the scientists, they needed some specialized databases to store this information. Yes, to store the information. And also they improved the tools. They improved the tools for data analysis also. So simply obtaining the DNA sequence, it was not enough. So the scientists, they needed some powerful computational tools for what? In order to store the data, compare the sequences, analyze it, and then identify the genes, and also to study the DNA variations. So this contributed to the development of a study, that is the bioinformatics. Okay, next one is the transfer technologies. Transfer technologies. So the technologies developed during this Human Genome Project could also be transferred to other areas, especially in industries and also in the field of biotechnology. Next is address the ethical, legal, and social issues. The short form is ELSI. So, for example, genetic information, it raises questions about privacy of genetic information and also use of genetic data and discrimination based on the genetic information and ethical concerns related to genetic testing. And next one is the coordination. So you can able to see here in the exam tip is coordination. So the project, it was coordinated mainly by US Department of Energy and also National Institute of Health. You can see here NIHS, and other countries like Japan, France, Germany, and China also contributed to this. So you can able to see the timeline here. So in 1990, the Human Genome Project was launched, and the project was completed in the year 2003, and May 2006, the chromosome 1 sequencing completed. So this chromosome 1 was the last of the 24 human chromosomes to be sequenced. So what is this 24 chromosomes? It is nothing but 22 autosomes and X chromosome and Y chromosomes. So next we are going to see how it was done, and we are going to learn the methodology. This is a most important topic in this chapter. Okay, so HGP, it used two major approaches. Let's see the first approach. Two approaches it was there. So the first approach was expressed sequence tags, that is ESTs. ESTs. So the first approach, it focused on identifying the genes that are expressed as RNA. Yes, identify the genes that are expressed as RNA. So when a gene is active, it can be transcribed to produce RNA, that we know. And then scientists, they studied these expressed sequences to identify the genes, and these are called as the ESTs. ESTs is nothing but the expressed sequence tags. So just think of a genome, yes, as a huge library, and some books are actually being read or used. So EST approach is to find the genes that are actively expressed. And what is the approach 2 then? Approach 2 is nothing but sequence annotation. So the second approach was to sequence the entire genome, and this included the coding sequence and the non-coding sequence. Coding sequence and the non-coding sequence. So after sequencing the entire genome, scientists, they identified different regions and assigned functions to them. What they did, they identified different regions and they assigned the functions to them. This process is called as the sequence annotation. And now what is the EST difference here is EST is find the expressed gene, and the sequence annotation is sequence everything and then assign the function. Yes, let's see this diagrammatic part. So this diagram, yeah, it represents the journey from the human cell to the complete genome sequence. So first one is the human cell. So the scientists, they begin with a human cell containing the complete DNA. So inside this nucleus, you can able to see the complete DNA. So the next one is the isolation of the DNA. So the total DNA is isolated from the cell, and we know that DNA is a very long molecule, so it cannot be sequenced easily as a one complete piece. Next one is DNA fragmentation. Third one, step 3 is DNA is fragmentized. That is, the DNA is broken into so many smaller fragments. Why it is done? Because DNA sequencing machines, they have technical limitations, right? And also cannot easily sequence extremely long DNA molecules. And the step 4 is the cloning. Step 4 is the cloning. So the DNA fragments are cut they are inserted into the suitable host organisms. So the commonly used host were bacteria and yeast. So here some special vectors were used. Yeah, special vectors also used, BAC, right? So they are bacterial artificial chromosome, and YAC, this vector is yeast artificial chromosome. And then step 5, they did the amplification process. So in the amplification, the DNA fragments are copied many times through cloning. What they did, the DNA fragments are copied many times through cloning, and this produces sufficient DNA for sequencing. And the next step is sequencing. So in sequencing, the DNA fragments are sequenced using automated DNA sequencers. They used automated DNA sequencers. So these machines, they work on the principle of the method developed by the Frederick Sanger. And next one is the sequence assemble. So the sequence assembly. So after sequencing, scientists have many small DNA sequences, right? So these fragments are arranged using their overlapping regions. For example, let us take fragment A as A, B, C, D, E, and the fragment B as D, E, F, G, H, and the fragment C as G, H, I, J, K. So the overlapping portions help them to arrange as A, B, C, D, E, F, G, H, I, J, K, right? And then next one is the computer analysis. So we have to analyze it. So the number of sequences was enormous, and humans, we could not manually align all the sequences. It's not at all possible. So specialized computer programs were developed to align and assemble the sequences. Yes, align and assemble the sequences. And next one is the annotation. So the assembled sequence were studied. Yes, and different regions were assigned a function. And next is the chromosome assignment. So finally, the sequences were assigned to the respective chromosomes. So let us see this. The first one is the human cell, and then the DNA is isolated, and the DNA fragmentation was done, and it is cloned into a bacteria or yeast, and it was amplified, and DNA was sequenced by Sanger method, and on the computer based alignment was done, and the assembly of overlapping fragments are done, and the sequence was analyzed, and at last the chromosome assignment was done. So these are very important thing. Yes, please watch the full video, then only you can able to understand. If you have any doubt, you can ask me in the comment section. So next we are going to see the salient features of the human genome. Now let us look at the important discoveries made from the Human Genome Project. What is the size of the human genome? First of all, the size is 3,164.7 million base pairs. And how much it was? Approximately 3.16 billion base pairs, right? And also, what is the average gene size? So the average human gene, it contains approximately 3,000 bases. So genes vary greatly in size. So the largest human gene is dystrophin. Yes, dystrophin. It contains approximately 2.4 million bases. And how many genes? So the total number of human genes was estimated to be approximately 30,000 genes. So this was much lower than the earlier estimates of 80,000 to 1,40,000 genes. And this was one of the surprising findings of the Human Genome Project. Next is the similarity among the humans. We know that approximately 99.9% of nucleotide bases are identical in all humans. So this means that all humans are genetically extremely similar. The small difference that remain contribute to the genetic variation among the individuals. And next one is the unknown gene function. So the functions of more than 50% of genes discovered were unknown. So this shows that knowing the gene DNA sequence, it does not automatically tell us the function of every gene. That is not possible, right? And also next is the protein coding DNA. You can see here. So 2% of the human genome, it codes for proteins. It codes for proteins. And this means that a very large portion of the genome does not directly code for the protein. Then repetitive DNA. So repetitive means it is nothing but repetition. So a very large portion of the human genome, it consists of repetitive sequence. These are nothing but DNA sequences that occur repeatedly. Sometimes it can be like hundreds or thousands of times it is repeated. And they thought to have no direct protein coding function, but they provide information. Now, what information they provide? They provide information on chromosome structure, chromosome dynamics, and also evolution. And next one is the chromosome 1 now. So it contains the highest number of genes, approximately 2,968 genes, and the Y chromosome contains the fewest genes. It is approximately 231 genes. And what are these SNPs or SNPs we can say? So these scientists, they identify approximately 1.4 million, yes, locations. So where the single base differences occur among the humans, and these are called as SNPs. SNPs are nothing but the single nucleotide polymorphism. Yes, and scientists, these SNPs help the scientists to identify the disease associated DNA regions, and also to study genetic differences and trace the human history. So this is the genome at a glance. So whatever I said, so everything again it is covered here. Yes, go through all these things. So let's have a quick revision of this Human Genome Project. So HGP is the Human Genome Project. So it was a mega project to sequence the human genome. It started in the year, it was that is launched in the year 1990 and completed in the year 2003. And the genome size was approximately 3.16 billion base pairs. And the genome number, it was approximately 30,000 genes, and it was less than 2% of the genome codes for proteins, and also 99.9 percentage it was of nucleotide bases are identical among the humans. And dystrophin, the largest known human gene, it had 2.4 million bases. And bioinformatics is a study. It is used to store the storage, retrieve, and analyze the genome data. And ESTs are expressed sequence tags. It is used to identify the genes that are expressed as RNA. Next is the sequence annotation, the process of assigning the functions. What is it? The process of assigning the functions to different regions of the genome after the sequencing. And what is this? BAC and YAC? They are nothing but bacterial artificial chromosome and yeast artificial chromosome used as vectors during the cloning of the DNA fragment. And SNPs or SNP SNPs, right? So they represent the single base variation among the individuals. So we have come to the conclusion part. Go through the full video. Don't miss it. We'll see you in the next video. Thank you.